I'll be talking about a little bit of data analysis in cancer and identifying some of the genes that we talk about in how they may impact cancer initiation and progression. Of course, I don't have any conflict of interest, and I kept two QR codes. This is for Apple and Android apps to do some analysis. When I go ahead with my talk, you'll realize what I am talking about, the analysis. But I work in cancer. I started working in cancer when I joined University of Michigan. And those are the days when the high throughput analysis like microarrays just started. So I started my work there and then, continue to look for genes that are changed in different cancers. Talking about cancer, of course, like Dr. Shreshtha mentioned, it's a epidemiological problem also. That is a disease that the cells starts growing in different organs, let's say lung or liver, and will not be controlled. They keep dividing, and there is no programmed cell death in these cases in aggressive cancers. Okay. And they can spread as well. And once they spread from like a localized organ to distant organ, it's called metastasis, and almost always is lethal. Patients will die. And one of the important things with this kind of cancers is a lot of changes happen. It is a continuous dynamic change at the molecular level. I'm talking about DNA, the RNA, the proteins, and all that. And that leads to cancer progression from early initiation to unregulated aggressive cancers. So these two apps basically, just to give a glimpse of different gene protein changes across different cancers, basically. As you all know, it's a big problem, not just in US, as you can see here, over 2 million cases identified, but across the world, particularly in some of the poorer countries, even the treatment option is very limited. So it's a global health crisis, basically. And as you can see from this American Cancer Society chart, there are a lot of varieties of cancer. Some of them are very prominent, like prostate cancer in men, breast cancer in women, and lung cancer in both genders, basically. And a lot of death also because of this cancer. This is US statistics. You can consider, think about the whole world as well. All this starts with the cells, right? We have cells, a lot of cells in the body. They estimate about 35 trillion cells in the body, distributed in different organs, the brain, the heart, the liver, all that places. And they always do the specific function where they have to function. In liver cells, they function what liver needs to do, right? Lung cells have different functions. So it is all done because of certain specific genes that are expressed and the other genes not expressed in specific organ. And there is about 20,000 genes, the protein coding genes they estimate. And there are other non-protein coding genes like long non-coding RNAs and microRNAs are also there. Okay. And of course, a lot of this message is in the nucleus at the DNA level. And the DNA can replicate and form more DNA and also transcribed into RNA. Messenger RNA is the one which codes for protein. Then the messenger RNA will code for proteins. There are 20 amino acids are in this protein chains. And once the protein is made, there will be additional changes that occurs, like adding of different groups to different amino acids, like phosphorylation or ubiquitination or acetylation and all that. So this creates a large variety of molecules in the cells, okay? And we are talking about 35 trillion cells in the body. And the good thing is most of the time, everything is, perfectly maintained, the gene expression, the replication, the transcription and translation. There are cases where, sometimes change occurs. For example, here, there is a blue cell, which is not normal cell. But what good thing is, if there is some mistakes, the so-called mutations and all, there will be a repair mechanism in the cells that will repair back that mistakes. Or there is programmed cell death. If the cells are mutated, there will be, those cells are killed. It's called apoptosis as well. But in certain cases, what happens is mutations can keep accumulating, and that can lead to early stage cancers and then very aggressive cancers. And like I said, when it becomes metastatic tumors, there's no cure actually. And what you see here in this list is different cancers have different progression time, actually. Some of the cancers are very aggressive. You always hear pancreatic cancer is very aggressive for that matter, 2-3 years of survival rates. Whereas some of the cancers here, it can go up to 5 to 15 years. There will be, depending on the diagnosis, there will be surgical removal or radiation therapy and a lot of treatment options also there. One thing I wanted you to remember is when we say lung cancer or breast cancer, it is not one type of cancer in every person's, okay? Because of this kind of molecular changes that can occur in different persons, it's heterogeneous, basically. For example, all these images you see from microscopes, these are all breast cancer samples, okay? But you see the architecture difference. That shows the heterogeneity at the even, morphological level in breast cancer in different patients. And if you look further, there are different mutations there listed here. So there are different breast cancer with different mutations. And this is just a highlight, but then there is a lot of gene expression changes, protein expression changes also in these cancers. Like I mentioned earlier, metastasis, when the tumor from local organ, like let's say breast or lung, goes into the blood vessel, moves to the distant organ, and then go and home in there, that is called metastasis. And that is like almost always incurable. But in the last 20-25 years, a lot of advances are made, and there's this concept of precision medicine or precision targeting is, came up in cancer, where individual patients that comes into the hospital will be sequenced to identify if there is any specific mutation and if there is specific drug for that mutations, right? So that has improved, the patient treatment, but still there is a lot to be done actually. So I started my work in this cancer genomics era during the days of microarrays. I don't know whether any of you heard. You can measure all the genes, gene expression using these slides, and each dot there you see is one gene and how much it is expressed, basically. You can always compare RNA from normal cells and hybridize with cancer cells, and they show up in different colors. And you can measure if it is higher in cancer, you will get different color like that. So around 2000, when I joined University of Michigan, Ann Arbor, they started a program to collect a lot of tissues from cancer patients, particularly their prostate cancer program is very strong. And we would collect this metastatic tumor samples from patients who just died. It's called warm autopsy. The body is still warm. We would collect all these tissues. And along with all the primary tumors and these metastatic tumors, we would compare the gene expression between all these tissues. For example, from benign prostate cells to early stage cancers, to aggressive cancers and metastatic tumors. So we measure all the 20,000 genes and also look for mutations by genomic sequencing. And then we analyze the data to find what are the major changes that happen in these cancers. And those are like about 25 years microarray. Now, of course, almost everyone does something called next generation sequencing, where you can actually measure not just the gene expression, you can measure the long non-coding RNA expression, microRNA expression, or identify mutations, SNPs, the variations, everything. So mostly people do next-gen sequence analysis nowadays. It become very cheap as well. And for protein changes, you would do mass spectrometry analysis in the labs. From microarray, we identified a lot of changes in prostate cancer from early stage. As the cancer progresses, we find there are different genes that are either downregulated or upregulated, and you can classify these genes are depending on their functions. Maybe some of them are cell growth factor receptors. Some of them may be cell addition molecules. Some are kinases and phosphatases. So we have a list of genes that identified in 2000 that are going up or down in prostate cancer. Similar work was done across cancers. Thousands of studies came out showing major changes across gene expression. And also protein expression. We did immunostaining on hundreds of prostate cancer tissues, showing some of these proteins also changed, increased or decreased in prostate cancer. And we continued this work, working on these genes, like showing its overexpression and its function in different cancers, and published a lot of targets. One of the things I particularly focused on is enzymes or cell surface markers. The idea is, probably you can treat this, enzymes or target these enzymes with small molecules. It is easier. And then also, if you have cell surface markers, you can develop antibodies to target them. I'll talk a little bit about one of the very successful target we worked on, EZH2, and also a transcription factor that is the first time we identified as a gene fusion in prostate cancer. And of course, in this last 20 years, this is a slide from like about 6 years. You see all these new drugs they identified for the targets. And also, some of these drugs which is already available, they identify subset of specific tumors where they can be treated with these drugs. This is the idea of precision medicine. So when you go to the clinics, they will find, any persons have these kind of mutations and then start treating with the specific drugs. However, the rate limiting factor is there is not enough targets or drugs that are developed. For example, this chart shows basically about 750 patients they are sequenced in a center, and they found only about 16 patients could be treated with these precision targets. So you see that large number of patients, they don't have targets. That's where cancer biology or oncologists need to come up with new, identifying new targets and develop new drugs, including pharmaceuticals, right? Of course, as researchers, we have our limitations. We cannot go and find new targets. We always like to work on a very well-established target and further study that. But now with all this data, there is a lot of opportunity to find new targets and potentially develop new drugs. However, all the data is there. How to analyze that data by let's say a biologist or a researcher in the lab. So that is a kind of roadblock. So what we decided about 8 years back is all this data, many of this high throughput sequencing data is deposited at different repositories, either at NIH, NCI, GEO, or Sequence Read Archives and all. If there are experts who can download the data and analyze, then only you can use this data. So what we decided was to collect all this large amount of data and then develop simple website, let's say, where you can click some button and start looking at various data points. That's what the motivation for us to develop this platform called UALCAN, University of Alabama Cancer Data Platform, basically. Of course, some experts helped in big time. Chad Creighton from Baylor College, he's a proteomic expert. So he analyzed all the proteomics data. Darshan was in my group. Now he's a faculty at Pathology. He analyzed all the RNA sequencing data and developed this platform, UALCAN, basically. Again, there is a QR code for that. And the detail about how we collected the data, how we normalized, and how we developed this platform is there in a lot of these papers. We incorporated proteomic data and all that. And of course, this has been 8 years now, and a lot of academicians and pharmaceutical people used. And we know that because sometimes we get appreciation for the effort, what we did. This is Jay Bradner, the then president of Novartis Institute of Cancer Research. Basically, he was appreciating our effort to develop and maintaining these platforms. And also, we keep a small world map where you see a lot of people using across the world this platform, basically, and highly cited also. That is a motivation to further add new data sets for this platform. So when you go to UALCAN itself, it shows simple buttons there. TCGA is for RNA expression. Proteomics, of course, for protein expression. And CBTN is basically pediatric brain tumor genomics data, RNA sequencing data. And then you start clicking the buttons. If I already told you that it's about 20,000 genes, and then various cancers you have. So you can either enter the gene name if you know gene that you want to look for, or just enter the cancer and just click the button there. That will give you the data about that genes. So you can add the gene name and then select whichever cancer you want also, or select whichever cancer from the left-hand side. Then if you select a cancer from the left-hand side, let's say in this case, I selected X gene fusions, and you will get something called heat maps. And you see there, you have normal prostate tissue and cancers also. And also what you see here is we classified it as X gene fusion positive prostate cancer. It's one molecular subtype of prostate cancer, and then X gene fusion negative prostate cancers. And you see there are differences at the molecular level. These are all different genes, okay? And individual line here is a patient. Of course, you can click on any of these genes and you start getting box plots showing how it's expressed, how much is expressed in normal compared to different classes of cancers. For example, here you see that gene PLA2 G2F is not at all expressed in certain subtype of bladder cancer at all, right? It's almost like normal, but subtype of bladder cancer, like luminal subtypes, have very high expression of this gene. Why you do this kind of classification is because let us assume there is a drug for this gene, PLA2 G2F, very good FDA-approved drug. You don't want to treat all these patients, neuronal basal squamous bladder cancer patients, with that drug because there is no gene expression in those patients. So that is the advantage of classifying all these gene expressions or protein expression to subcategories. It may be molecular subtypes, histological subtype, or based on race or whatever the parameters the study provides. And sometime when we created this web platform, a lot of people ask us, hey, we have, oncologists, for example, this case here, renal cell carcinoma. Kim Rathmel, if you know, she's now NCI director. She had subdivided renal cancer into clear code types, CCA and CCB subtype. She asked, can we provide that kind of subclassification in UALCAN? That's what we provided here, for example. And according to her classification, the CCB subtype is more aggressive, and which you find in UALCAN that the specific gene in CCB subtype is increased and shows poor survival. This is a Kaplan-Meier survival curve that probably you all know about these curves. And then, of course, when you look at gene expression as biologists, we also want to look at how it may be regulated in cancer. So we provide something called co-expression analysis. Here it gives Spearman correlation. So you see that if there is high level of PGK1 gene expression, you also have LDHA gene expression increased. So it gives a slight idea that maybe one of the other gene is regulating the second gene. So you can go to lab and test this by either removing one gene and looking at the other gene. And as such, as cancer biologists in the lab, we can do this experiment very easily. It's not complicated. If you have a normal cell, you can add a gene and see if they become cancerous. And if you have a cancer cell, you can just remove the gene. These are all very easy technologies nowadays. And then see if the cancer cells start dying or stop proliferating. So it's not complicated to do the cancer biology work on any of these genes. And of course, TCGA tried as much as to include a diverse population in their studies, but they don't have much African-American or Hispanic patient data. But whatever they have, we try to provide that classification also. For example, you see 22 patients, we have African-American patients, and in this specific bladder cancer, it's slightly higher expressed, this specific gene. I must add, there is no normal controls for African Americans. It's just the relative expression increase in African Americans compared to American patients compared to Caucasian patients. Apart from this, just the expression, you can do a lot of other analysis using UALCAN, like you can look at the survival analysis. You can also look at something called promoter methylation analysis. So promoter methylation is a type of gene regulation. So it's generally thought if there is the promoter of the genes are hypermethylated, gene expression is reduced. So you can do that kind of analysis. Like I showed, correlation analysis you can do. And also you can look at a specific gene. If you're working on breast cancer, you can look at a specific gene that you are working across all the cancers. And we also provide many external platforms that is very useful for researchers. Like GeneCards tells a lot about whatever that gene you are working on. PubMed just wanted to show specific gene, how many publications are there in, for that gene in that cancer. And then Human Protein Atlas provides high resolution pathology images of all this staining of these genes in cancers. Like that target scan tells if there is any microRNA that regulates your gene of interest, okay? GTEx is a repository where you see the gene expression of every gene across all the normal tissues in human body. And then you have also drug bank where you may have certain drugs that targets gene of your interest. And then you can either test that in the lab or think about if it is FDA approved for, let's say, diabetes or Alzheimer's, you can think of targeting cancer if that specific gene is very highly expressed in specific subtype of cancers. And of course, you can add all these genes and get different type of heat maps. You know, all this you can download and then download as JPEGs or PDFs or SVG files, and then use it in the grants or publications also. All the download button will be on the right-hand corner in the top 3 lines that will allow you to download the figures. This is a example of pan-cancer analysis of a gene called GLY80L1. I just showed this because you see many of the cancers is not overexpressed. Blue is normal and red is cancer. But you do see slight overexpression of that gene in prostate cancer. Red is cancers, blue is normal. So when I looked at the Human Protein Atlas for that gene, you do see increased staining of that protein in the prostate cancer. And then I can, of course, look at the bigger images from that web platform, the Human Protein Atlas, and you do see increased immunohistochemical staining of that protein in prostate cancer and not in the benign glands, showing that, how you can integrate all this various data to make, a case for, let us say, GLY80L1 as a biomarker or target. And I mentioned about promoter methylation. I mentioned that, many of the genes that are silenced are hypermethylated in the promoter region. For example, here is the gene SOS-TDC1. Its promoter is hypermethylated here. And you look at the gene expression, it's downregulated. This you can go back, into the lab and then validate. Yeah, indeed, this gene is maybe downregulated in prostate cancer in this case. And then just like gene expression, you can look at protein expression also in the proteomic tab. It's important to know that, all the gene expression changes will not be reflected in protein expression changes. Sometimes gene expression may be higher, but protein may not have changed. And there are additional regulation of proteins also. So that's why, it's important to integrate all this data. In this case, we are showing P4HA1 protein, which is increased across different cancers. We in the lab itself, we did validate this using immunohistochemistry, particularly in prostate cancer, and then also understand the biology of this protein. Apart from protein, we also have gene expression data for something called long non-coding RNAs. It's a new class of molecules. They don't code for protein, but they're long mRNAs. And they're still trying to understand how they regulate the biology of cancer cells. Not very well established. Different long non-coding RNAs have different kind of functions. Then there is microRNA, which is really well established reasonably. And in fact, this year, Nobel Prize went to those guys who identified microRNAs, Gary Ruvkun and Victor Ambrose. So these microRNAs are tiny molecules, and when they are mature, they're about 22 nucleotides. And what the early prediction or early studies showed is that they can bind to the 3' UTR, untranslated region of genes, and block protein synthesis. Now, how a researcher can use this data to validate a target or biomarker and then develop drugs, or use this information for treating patients. I'll show an example here about a gene called frizzled-8. It's a frizzled Wnt receptor, basically, and there are drugs that are in experimental trials for this gene. what we saw from using all this data is that there is a gene fusion in prostate cancer called ERG gene fusion. ERG is a transcription factor, okay? You cannot easily target that. And we showed about 50% of prostate cancer had this gene fusion. So then you can look at the co-expressed genes that goes with the ERG, and we found that one of the genes, frizzled-8, because we looked which are the genes here have drugs. Frizzled-8 had drugs. Then we thought we'll look at frizzled-8 a little more closely. And what we saw was that, ERG is overexpressed in prostate cancer. Similarly, frizzled-8 also is overexpressed. But what is interesting is that in only ERG gene fusion positive prostate cancer, you have ERG gene expression. This is transcript per million. And all other prostate cancer, you don't have ERG expression. Interestingly, similarly, frizzled-8 is overexpressed in only ERG fusion positive prostate cancers, right? And like I mentioned, we showed earlier that about 50% of prostate cancer had this gene fusion, where ERG is fused to an androgen-regulated gene called TMPRSS2. And human prostate is an androgen-regulated organ. And whenever there is androgen, it would drive this gene fusion and ERG expression, which in turn we showed that increases the expression of frizzled-8. And like I mentioned, there are a lot of drugs that is trying to target frizzled-8. And if they become FDA approved, you can think of treating about 50% of prostate cancer with these drugs for frizzled-8 potentially, right? With the success of this platform to distribute the data across the world, we also thought we can now go deep into certain specific cancers, particularly common cancers. So a student, Santosh, joined our lab, and we decided we develop a breast cancer focused portal where we will have not just the TCGA, but studies from across the world. So we so far collected about 20,000 breast cancer patient samples. And also in this study group, we have patients treated with different drugs, the breast cancer patients. We also collect a lot of single cell sequencing data, which is a new data set that is coming, where they would isolate from the tumor microenvironment, epithelial cells, endothelial cells, fibroblasts, all those separate them out, sequence. So that's a data set also we included. And of course, the backend involves a lot of information. And a publication that describing this is in review in Nature Breast Cancer, and hopefully it will be accepted soon. Like I mentioned, we provide all the analysis that I mentioned in you all can, and also we provide this kind of information where you can find the gene of your interest, where specifically, which cell type it is expressed. It is important to know, right? As biologist, or if you're targeting, you want to understand where it is going to be targeted. So you can get this kind of graphics, again, download it and use it in your publications. And like Dr. Sadeep mentioned, we are also creating a head and neck cancer specific database where we'll collect tons of data from head and neck cancer patients from across the world, deposit that, and create this platform where you can again find subclass specific gene expression. For example, in head and neck cancer, HPV positivity and negativity is important, and we will provide that information for every genes, and also survival information for the patients. Apart from that, we'll of course include all the available single cell sequencing data as well for head and neck cancers. Apart from that, people are always in the mood these days. We created this mobile app. It will not provide all the utilities that we have in the web platform, but you can do a lot of gene expression, protein expression analysis using this mobile app, which is free to download, basically. I'll quickly tell how, as biologists in the lab, at UAB or anywhere, what we do once we identify a gene. So one of the most successful work from our group and others are this histone methyltransferase called EZH2 gene, okay? So we identified EZH2 as kind of overexpressed gene in metastatic prostate cancer, particularly here, red represents increased, green represents low. So we saw that EZH is high in prostate cancer. Of course, nowadays people worked on it extensively, and as you see, it's actually overexpressed across all the solid tumors. So once we saw this EZH2 is overexpressed, I'm talking about early 2000s, we went on to test that on large tissue microarray where we had patient sample from over 200 prostate cancer patients. That's each dot there is a tissue from individual patients. And we did immunostaining to look at the protein expression. EZH2 is a nuclear protein. And we saw there is increased protein expression. This is the quantification by pathologists. So the EZH2 protein expression increases with disease aggressiveness. And also when we looked at the failure-free survival, if there is increased EZH2, patients they don't survive long. In this case, PSA or prostate-specific antigen comes back. Whereas if it is weak, there is no PSA recurrence. Similarly, we looked at EZH2 in other cancer, particularly breast cancer. As you see, in subclasses of breast cancer, the triple negative subclasses, which is the most aggressive, shows high expression of EZH2. Again, we went on to validate with pathologists the staining, in the protein staining, and you see that there are some breast cancer with very high EZH2 protein. Just like prostate cancer, if there is high EZH2, patient survival was poor. And like I said, we can then go to lab and do some simple experiments. So in this case, what we did was we put EZH2 gene into a normal breast cell and test what happens to them. So there is no EZH2 here. This is a control. And here there is EZH2. And then we do some sort of assay where cells, whether they move through the membrane, okay? It's called invasion assay. And if you add EZH to normal cells, the cells start moving. And similarly, we do multiple experiments to show that EZH2 is oncogenic or causes potentially cancer. And then we wanted to look what exactly EZH2 does. So we added EZH2 to normal cells here and looked at all the gene expression. What we saw was when you have EZH2, all the genes are downregulated. And when we closely looked at all those green genes, many of them are a class of gene called tumor suppressors. You can consider these as good genes. Good genes are downregulated if there is high EZH2. Of course, when we published that, another group showed that it's an enzyme that modifies histone, one amino acid of a histone H3 tail, okay? Basically, it modifies adding methyl group to a histone K27 in histone tail H3. So there are multiple histones. So that's one modification where it adds 3 methyl groups, makes the chromatin condense or compact. And what happens because of that is now the transcription factors cannot go and bind, whereas open chromatin transcription factor can bind and increase gene expression. But if there is EZH2, chromatin is condensed and genes are not expressed. That's why the gene expression was downregulated when we add EZH2. And we tested some of the genes, that green genes that I showed in the previous slide. For example, we looked at E-cadherin, which is an important gene. It's involved in cell-cell addition, and it's a cell membrane protein. And here you see when we add EZH2, that's a green color, you lose that membrane addition protein, and then cells start becoming invasive, basically. And we can actually do in a mouse experiment where you remove the EZH2 from cancer cells and inject to the mouse. And you see that if there is no EZH2, you don't see the tumor growth at all. And again, as biologists, we want to see why EZH2 is increased in the first place in cancer. So we looked at a lot of options. If there is a deletion of increase or amplification of EZH2 copy, which we did not see. And we thought about potentially microRNA negatively regulating it. So when we tested that, of course, there are tools, prediction tools you can test, and we found that microRNA 101 may be regulating it. So when we use that microRNA 101 in cancer cells where there is high EZH2, adding microRNA 101 blocks EZH2 expression, okay? And siRNA is a chemical compound that also blocks EZH2 expression specifically. So that shows that microRNA 101 downregulates EZH2, but in cancer, that microRNA is basically downregulated. Because microRNA is downregulated, you see increased expression of EZH2. And of course, EZH2 is now extensively studied in some lymphomas. It is mutated and it's activating mutations. So a lot of people wanted to target this gene. Okay, we did ourselves do the screening for about 8,000 small molecules and developed a specific, reasonably specific inhibitor. We don't know how, target, other non-specific interactions, but what we had was that specific inhibitor, reasonably specific inhibitor, downregulated the tumor growth in mouse tumors. But by then, a lot of pharmaceuticals were interested in developing highly specific, easiest to inhibitor. And as you see, all this publication from pharma, they developed very, very specific inhibitor targeting easiest to. Now we use this kind of reagents from different companies, basically, to do our in vitro studies or in vivo mouse models. Other thing we did recently is looking at how easiest2 can be used in different cancers. Easiest2 forms a complex called polycomb complex, which actually downregulates the gene I showed in the previous slide. There is an opposite complex called SWI/SNF complex, which activates the gene. So what I'm showing here is in about 25% of bladder cancer, this ARID1A, which activates the gene, is mutated. It became inactive. And in those cases, if you block EZH2, it's called synthetic lethality. You can potentially see cancer cell not growing. So that's what we wanted to test. And incidentally, you see that these two are ARID1A mutant cells. When we add EZH2 inhibitors, the tumors don't grow. Whereas these are all ARID1A wild type cells, and tumors go even with EZH2 inhibition, showing that this is a synthetic lethal combination that can be potentially used in about 25% of bladder cancer patients. We did this in another model called patient-derived tumor xenograft models. There also we saw that if you add EZH2 inhibitor to this PDX with the ARID1A mutation, you see reduced tumor growth compared to the ARID1A wild type tumors. Okay. So in conclusion, we can use a lot of this public data through different web platforms, including our web platform. You all can to identify specific targets. And we did a lot of biology work on different targets. EZH is one of them, for which now there is FDA-approved drugs. And of course, integration of all this data is very important, in understanding, developing new target, understanding pathobiology, and finally, effective treatment of individual cancer patients. With that, I would like to thank a lot of people involved in the bioinformatics side of things. And, I would like to thank Sadeep also for supporting head and neck cancer platform development, Siddharth Kumar for developing this mobile app. Remember, I can't code a single line. So these are the people who develop these things. I just give the ideas of what we need to see. And of course, proteomic data was done by Chad Creighton from Baylor. Chang Pan from Harvard did the PDX model for bladder. And Guru Sompaude, who was at UAB once upon a time, he helped with the bladder cancer studies, not just ARID1A, but several other bladder cancer studies we did. He's an oncologist. A lot of the early work on EZH2 was done with my postdoc mentor, Arul Chinayan, and Selena Claire, who is a breast cancer pathologist. And also, I'd like to thank all the funding support.